And first of all, thank you for your participation in that session. Before starting, I'd like to remind that because of tight schedules, every presentators please keep just 20 minutes for your presentation, including question time. And so, first of all, I would like to explain what purpose of this session. So, in this session, I asked every presenter to introduce their own work, especially for informatics or data management. So every participant will know what they do by informatics or database construction or data analysis. So, and I invited 4 presentators, and also I myself. The topics are like that. So, first presentation is from I, and the title is the 9th Rice Annotation Project Meeting. I apologize that this number is slightly different from the real IRGSP meeting, but this is the 9th RAP meeting. And the content of my presentation is like that. There are 3 parts. At first, I would like to introduce our project, Rice Annotation Project. And second, maybe everybody wants to know the latest version of the Nippon Valley genome. And finally, I summarize the current activity of RAP. And this is an acknowledgement. Actually, Rice Annotation Project is managed by our institute, National Institute of Agrobiological Sciences, and Dr. Takeshi Ito is main manager. And also to construct the latest version of Nippon Valley genome, Dr. Burrell's group from Michigan State University kindly support. So, I think everybody already knows, but IRGSP, International Rice Genome Sequencing Project, was launched in 1998, and in 2004, version 3 of IRGSP genome was released in public, and this paper was published in Nature next year. And after that, to provide high-quality annotation of rice genes based on IRGSP genome, the Rice Annotation Project, named RAP, was launched, organized in 2004. And from that, first RAP meeting was held in Japan, and after that, until this year, in total of 8 meetings were held in variable countries. And to release the RAP annotation data, we developed RAPDB, and until now, 4 times updates were done. And our concept is to curate all functional descriptions assigned by automated method. It means manual curation for all rice genes, if possible. And also, this gene structure or annotation is focused on gene loci, validated by whole-length cDNA clones. And this is a rice annotation standards, and it's defined and described in the Susan McCouch's papers published in Rice Journal in 2008. So RAP followed these papers and that concept. And to curate the many genes manually, so RAP held 2 times of manual curation jamborees in Japan. In Lapua meeting, more than 70 participants joined, and lap 2 around 30 to 40 curators joined. And every curator sat down in front of the computers for several days from morning to evening to curate, check the automated description of genes, and then by checking literature one by one, and finally they revise their annotation data. So here I just showed one example. So this gene is automatically predicted a certain ORS, but no description, just conserved hypothetical protein. But after checking a certain manuscript, these genes are seed dormancy 4, SDR4, is quite important genes. So without any this manual creation, for every user, whether these genes are important or have a certain functions, nobody knows. So that's why we believe that this kind of manual creation is quite important for further analysis. However, there are a big problems for rice genome and that annotation. There are 2 standards for rice genome. So one is IRGSP genome and our lab annotation data, and second one is Tiger MSU groups data. Many of users didn't know that the should molecules rice genome structure are different between IRGSP and Tiger version. To solve this problem, we collaborate with each other, and then by using a new method termed optical mapping and evaluation by NGS data, we constructed the latest version of rice genome named OS Nippon Valley Reference IRGSP 1.0 last year. Still, there are 2 different annotation data, but it's quite, we don't integrate into one because this kind of secondary data is quite useful for every user, we believe. And here, by using 2 slides, I explained how to revise genome sequence by NGS data. In case of Illumina reads mapping, because of the many read mapping results, we found that this kind of SNPs-like errors. So in this case, on the reference genome, this site is adenine, but all or almost mapped Illumina reads shows cysteine. In this case, we revised this site from adenine to cysteine. In total of around 5,000 errors, we're not sure whether these are real errors or not. But when we used majority of this kind of sites, around 5,000 SNPs or indels were detected, and in this latest version, they are revised. In case of 454 reads, we use this data to detect slightly longer longer errors of indels. Only 5 large indels were detected and revised by using 454 reads. By using genome sequence, we annotate 35,679 protein coding loci with transcript evidence, including within and cross-species transcript evidence. And after manual curation or literature curation, 1,600 loci are assigned by gene symbols, like SDR4 and HD1, like that. And now, from the LabDB G browser, users can see the expression profile based on RNA-seq data like that. And of course, curated annotation data is available. And also, we developed another databases for gene family and phylogenetic trees. And also, recently, we released the NGS data viewer. And also, now we are developing SNP viewer to integrate the variable accession lines data. As a summary, so now LabDB tried to provide not only the curation data, annotation data, but also these kind of secondary data to facilitate experimental research not only in rice, but also other crop species. Thank you for your attention, and any questions are welcome. Okay, so I would like to move to the second presentator.